Good day, everybody. I'm very proud to make a contribution to the Science Cafe with a poetic song about my own experience with the green environment in my beautiful city. The greening of cities, it's a subject I adore. 3 phenomenal female speakers in the house, need I say more? Maria, Mary, Helen, they are great in their own way. I googled them, curious what they were going to say today. Helen, will she talk about creating parks and planting trees? And Mary about pocket prairies and foraging bees. Maria, I assume, will warn us firmly from overseas about the risks of ticks and all kind of tick-borne disease. I was looking forward to their stories from the start. As a preparation, removed the tiles in my backyard. Created an environment for birds and honeybees, where they could live in peace, hang out in flowers and in trees. It felt like a success when I was doing this in March. My backyard within a week looked just like Noah's Ark. Exceptional species, rare and never to be seen. They came in pairs, surprisingly, appeared amidst the green. I welcomed them with all my heart, couldn't believe my eyes. 2 birds, 2 bees, 2 beetles, and 2 yellow butterflies. My Garden of Eden seemed to have a lot to give. But not only the insects found their way to where I live. A shelter, a safe haven, an oasis I created, where pets like dogs and cats and deers and lizards met and mated. They came from east and west, all species came from everywhere, and shared my secret garden until the space became too rare. 2 foxes, 2 mosquitoes, and 2 wolves, they came along. Beautiful and more than welcome here, don't get me wrong. The zebras and the snakes and the macaques had a ball. It got a little crowded, but there was still room for all. The crops, they grew like hell, thanks to the big raccoon's poo. Yes, everything seemed fertile in my precious little zoo. Comfy on my terrace, and the view is more than grand. 50 shades of grey, thanks to the female elephant. Next to me on the ukulele, Mr. Ilko Minkveld, ladies and gentlemen. The lions came, the bats, and other new beasts on the block. The ladies, they inspired me before I heard them talk. I wonder what they have to say, and I just wanna bet they share my enthusiasm about the love bites of the bat. Mrs. Gardener, Mrs. Wasser, and Mrs. Esser, go along. My flock needs to be fed, so I am finishing my song. We will learn from you without a doubt, your stories will stick. It's that green world you're gonna paint that makes us all tick. Good luck to the 3 of you.
Welcome to this Science Cafe today. Unfortunately, we can't meet in person with most of you, but I'm really very happy that there is a new edition of this Science Cafe. And hopefully very soon, when vaccination coverage increases further, we can meet again later this year. For those who have missed it, well, the NCOH is the Netherlands Centre for One Health is still alive, and we will continue the coming 5 years in a new period between all the different academic centers that collaborate within the Netherlands Centre of One Health. And we're also drawing up plans for that coming period. In particular, we are working on a pandemic research program And tomorrow the executive board will meet and discuss this further. But for now, we'll focus on green cities and livable cities. And because the introduction of more green space and also blue space introduces all kinds of One Health issues. I'm very glad that we have a good program with 3 distinguished speakers. And I hand over the word to Arjan Stegeman, who will introduce this afternoon further. Arjan, please go ahead.
Thanks a lot, Dick. Well, my name is Arjan Stegeman. I'm normally a professor at Utrecht University, but it's really a privilege for me to be your host for this webinar. And I think, as Dick indicated, we have a really exciting subject to deal with this afternoon. Because for those of you who have seen other webinars of NCOH, because there have been a couple, mostly they are focused on diseases being transmitted from animals to humans. So these would either be farmed animals or wildlife has been a topic of webinars. But this time we really have a different subject, the One Health challenges of greening cities. And of course, many of us live in cities. I myself live in a city, and we know from history also that green in the city has been a topic already for I think for hundreds of years and even longer. And we can still see that in many of the cities by many of the old parks that are present there. But now nowadays it has gained new interest. People are waving here. That's why I get a little bit distracted. But now it's getting more interest also because of climate change, the greening of cities to at least absorb carbon dioxide, cities helping in increasing the biodiversity. And also, we know that green in the city makes cities more attractive. So people like to live around green environments. And of course, these attract animals, insects, and all kinds of others, living beings, which is also interesting to look at from the One Health challenges. And from that perspective, I think we have 3 exciting lectures here, talks on this subject that I will gladly start introducing. But before I do that, although I'm the moderator, I am actually in really treating the guests that present here well deserved. I'm really depending on your help. So please use the chat in the YouTube, as this is broadcasted live. Please use the chat. If you have any questions regarding the speakers, send them over here. They will end up here on my screen, so I can ask them. And in that way, you can also actively engage in this webinar. And I would really appreciate that. And I'm sure the presenters would also appreciate that. Since we're short of time, I stop the introduction here, and I will want to introduce our first guest, our first speaker here, who is Dr. Helen Esser. And Helen Esser, I'm really glad you're here, and you're live here. That's even more special. She's an assistant professor at the Wildlife Ecology and Conservation Group at Wageningen University. And as a disease ecologist, she studies ecological interactions between arthropod factors such as ticks and mosquitoes, the pathogens that they transmit, and the vertebrate hosts on which they feed. Her ambition is to understand how environmental changes can trigger disease outbreaks by identifying the ecological mechanisms of these diseases. And the title of her talk is Biodiversity, Benefits and Health Risk. Helen, the floor is yours.
Thank you, Arjan, for this introduction, and thanks to the organization for inviting me here today. Very glad I could be here. So, the picture that you see here is the vision of Italian architect Stefano Boeri of how future cities should look like. And as you can see, it's like cities that basically blend in with nature. Now, before you think that this is science fiction, this exact city is currently being built in China. And many other cities around the world are currently embracing the idea of urban greening. So what is urban greening? Basically, urban greening is about incorporating vegetation into a city's design and landscape in order to create mutually beneficial relationships between city dwellers and their environment. Now, this mutually beneficial relationship is key here, because that might not always be the case. So that is the topic of this talk and the next. But before we dive further into that, why is urban greening currently such a hot topic? Well, at this moment, more than half of the world's population live in urban areas, and increasingly so in very highly dense cities. And as you can see in this picture, in this graph, by 2050, it's expected that over 2/3 of the world population will live in urban areas. This poses a lot of challenges, because unfortunately, many of these cities are plagued by urban health hazards. Think of pollution of air and water, heat island effects, and of course, infectious diseases. Some cities, like Beijing, as shown here, are even expected to become unlivable by the year 2050. One solution to these problems is urban greening. Now, examples of urban greening include traditional city parks, as you can see here below, the Vondel Park, but also living walls, or rooftop gardens, or even vertical forests, like this green tower that was built in Milan by the same Italian architect. And these urban green spaces provide lots of health benefits. First of all, oh, something is wrong with the letter type, excuse me. First of all, they help with climate change adaptation. So, for example, they help keep cities cool by preventing heat island effects. They improve air quality, for example, by absorbing and reducing pollution. They also help with carbon sequestration, so basically absorbing CO2 from the atmosphere. They also retain water in times of drought, while absorbing water during excess rainfall. Now, both of these, droughts and excess rainfall, are predicted to become more common in the future climate change scenarios, at least in the Netherlands. Moreover, studies have also shown that urban greening improves the physical and mental health of humans. On the one hand, by stimulating sports, but also by lowering cortisol levels. People who are surrounded by green are usually more calm and relaxed. Now, urban greening also increases biodiversity, and this in turn brings other ecosystem services, such as pollination by bees. Now, that will be the focus of the third talk today by Mary. And as a disease ecologist, I'm also specifically interested in this biodiversity aspect. Specifically, how will an increase in diversity of wildlife hosts affect the density and infection prevalence of arthropod vectors, such as ticks and mosquitoes? And how will that, in combination with human exposure, affect zoonotic disease risk? Now, there's ample evidence that high biodiversity could decrease the infection prevalence through a so-called dilution effect. However, evidence of the opposite also exists. So high biodiversity could also increase infection prevalence through a so-called amplification effect. However, most of these studies were conducted in natural ecosystems, such as forest and forest fragments, and it remains unclear how this relationship is, or how it looks like in urban green spaces, because urban green spaces have been relatively little studied. So that is an important question. How will urban greening affect, and hence the increase in biodiversity, the ecological relationships between wildlife, vectors, and pathogens? And how does that, in combination with human exposure, affect zoonotic disease risk? For example, predators that you could invite into the city if you have increased biodiversity are important pest control agents, and they could lower infectious disease risk by lowering or decreasing the density of important disease reservoir hosts, such as rodents. But one may wonder how effective they will be in reducing rodent densities if there's an easy snack to be gotten here in the trash bin. So these are questions that are still unanswered. In addition, bringing in extra wildlife can also bring in additional parasites. Many predators are themselves reservoir hosts for parasites and pathogens. For example, in the case of foxes, they are an important reservoir host for a tapeworm called fosse lintworm in Dutch that can be transmitted to dogs and humans. And if left untreated, it can even be fatal. Another thing to consider is that urban green spaces or increased urban green spaces could also increase movement of wildlife, either between urban green spaces or between urban green spaces and nature areas. and this could facilitate the movement and spread of parasites such as ticks. Now, deer are an important factor to consider here, because deer, both in the US and in Europe, are the most important host for the adult female ticks of medically important tick species. And so on the one hand, if you have lots of deer, you will probably also have lots of ticks. At the same time, however, deer are incompetent hosts for example, the Borrelia bacteria, which causes Lyme disease. So if there are lots of deer, there could be the situation that the deer divert tick bites away from competent hosts, such as rodents, and hence decrease the infection prevalence of ticks. So it's kind of a dual role that these deer have. On the one hand, they increase tick density, while on the other hand, reducing the infection prevalence. And this may result in nonlinear relationships between deer density and the density of infected ticks. Now, I'm sure that Maria will talk more about this in her next talk, as she has done exactly the type of study that we would like to do here in the Netherlands, so in the effect of urban green spaces, wildlife hosts, and tick-borne disease risk. But for now, what is the situation in the Netherlands? Well, in contrast to the US, tick bites in the Netherlands are mainly associated with forest ecosystems. And deciduous forests in particular have high tick densities. This is on the one hand because these forests host a very high density and diversity of wildlife that the ticks use to feed on, but they also have the right microclimatic conditions. The most common tick in the Netherlands, Ixodes ricinus, is very sensitive to desiccation and requires a minimum of 80% relative humidity to be able to survive. Now, forest provides the ideal habitat for ticks. However, very green city parks like this one in Rotterdam with lots of trees and probably birds and mammals could also provide the right habitat for ticks. So definitely you will find ticks here too, albeit in lower densities. If you have a very green garden, you might also have ticks. So a garden like this, which has plenty of space for ticks to hide and then not desiccate, birds, mammals that visit, for example, if your garden is close to a forest and looks like this, and these roe deer might visit your garden, you could potentially have ticks in your garden as well. However, most Dutch gardens do not look like this, but unfortunately more like this. This is not the type of garden where you will find ticks. There's not enough wildlife, the ticks will desiccate. So then the question is, how common are ticks in Dutch gardens and green spaces? Well, one study has shown that 30% of people reporting tick bites believe that they were bitten by these ticks in their own garden. 30%. So that is a lot. However, it's important to realize that we don't know what type of garden these people had and how urbanized, or conversely, how green their environment was. And also whether these people were actually exposed to the tick in their garden or whether they discovered the tick bite while gardening. So there's still lots of open questions here. And as you can see, city parks account for very little, like 2.5% of tick bites. Another question that remains open is whether ticks are able to maintain viable populations in urban green spaces in the Netherlands, for example, gardens or city parks. It's possible that ticks are able to complete their life cycle on, for example, hedgehogs, rather than on roe deer. On the other hand, it's also possible, if that is not possible, then the ticks are dependent on continuous introductions from nature areas where roe deer live by, for example, birds that migrate between these nature areas and urban green spaces, or pets, like when you walk your dog or your cat walks around the neighborhood and visits nature areas, they can bring in ticks. But this is something that we do not yet know. So currently, we're writing a grant proposal to study these questions and others, and particularly we're interested in knowing how urban greening affects tick-borne disease risk in Dutch cities via its effects on wildlife, ticks, and people. We hypothesize that urban greening and its biodiversity could, on the one hand, increase tick-borne disease risk. First of all, urban greening provides more suitable tick habitats, which can in turn increase both the survival and questing behavior, so basically the host-seeking behavior of ticks. more urban green spaces also provide more habitat for hosts. So higher host density might result in higher tick densities. And as I previously mentioned, urban green spaces could also increase the movement of host animals that carry ticks and pathogens. And finally, urban green spaces can increase recreation by humans. Think of sporting, picnicking, hiking, walking the dog, and hence increase human exposure to ticks. At the same time, however, you could also hypothesize that urban greening could decrease tick-borne disease risk through the so-called dilution effect. So basically, a higher diversity of hosts could lead to a dilution of infection prevalence by diverting tick bites away for more competent hosts like rodents. And also, as previously mentioned, perhaps predators could reduce reservoir densities. As I said, also this remains to be studied. And then finally, animals such as birds and mammals that live in cities and have plenty of food available and maybe less predation risk could have a more stronger resistance to infection or to parasites, which could then lower disease risk. So these are things that are still open questions, particularly in the Netherlands. And as I've mentioned, Maria will talk more about this in the next presentation. So I would like to finish by saying that it's important to realize that parasites and the pathogens are an integral part of biodiversity. So if you increase biodiversity, it's also likely that you will increase the diversity of parasites and pathogens. At the same time, high biodiversity through urban greening provides many other health benefits that may outweigh these potential other health risks. Now, if we're talking about urban greening and the many health benefits, we should not, however, neglect these potential health risks that they bring. We should inform the public so that we do not lose this, basically the common, the acceptance of urban greening. So with that, I would like to end this talk.
Thanks a lot, Helen, for your presentation. Well, I can applaud alone, but the others can do it on the other side of the screen. You just will not hear it. Excellent talk. Well, some questions came in. One is, of course, you advocate that we need this biodiversity also from the parasite point of view, but still people tend to find it nasty when they get tick bites. So the question is, when they have their gardens and the woods, you're not very fond of paving these gardens entirely. What can people do to kind of manage the tick burden in their gardens?
Yeah, that is the question. That's really difficult because it's kind of a how would you say that? Kind of a trade-off. If you make your garden more suitable, more attractive to wildlife, for birds to visit, for insects to come, you will inevitably also have a higher chance, I think, of birds coming in with ticks. So really the introduction then of ticks is something that you cannot avoid. And then regarding the...
But is there anything they can do without putting the entire garden full of insecticides or paving it?
I don't think so. It's really, in the end, it always comes down to check yourself for ticks every day if you've been in the green. That's in the end the one important thing that you can do.
Okay, so it's a risk. It's the risk of nature, and of course that's true. One final question, because we're short of time, but you showed us the nice picture of the deer in the city, and of course today it was in the news that in China they also had elephants straying into the cities. So how would this greening of the cities affect those movements? Because I think there are some animals that really bring more danger than others.
Yeah, so several studies from both in Belgium and the US, actually Maria's study, have shown that if you increase urban greening and the connectivity between these green spaces, then you increase the movement of these animals, and that could negatively affect health risk. But Maria will tell more about that.
Thank you. You nicely bridged that for me. Thank you very much, and thanks again for your excellent talk, Helen.
Thanks for inviting me.
So that brings me immediately to our next speaker, who unfortunately cannot be here, but will talk to us from the United States. Your name has been mentioned already, so you're already famous here in this webinar before having talked. Maria Duke-Wasser. She's a professor of ecology, evolution, and environmental biology at Columbia University. and she's interested in environmental and anthropogenic factors driving the emergence of vector-borne and zoonotic diseases. And during the next 12 minutes, she will give a talk entitled The Role of Urban Green Space Connectivity on the Emergence of Tick-borne Diseases. And please, public, send your questions through the chat so that we also can ask Maria some questions. Please, the floor is yours.
Thank you. Is my screen showing okay? Hi, yes. Can you hear me? Can you see my screen okay?
Yes, excellent.
Okay, thank you so much. Thanks so much for inviting me. I'm very excited to be here, virtually. And so I'll talk today about some of the work we've done and some of the ideas. It's ongoing work on urban green spaces in the United States, in particular in New York City. So Helen has given a great introduction. So a lot of the introductory topics, I'll go a little quick, which is good because she already talked about them. So Lyme disease is the most prevalent tick-borne disease and really vector-borne disease in the United States. And as Helen mentioned, there's two aspects to human risk of Lyme disease. There's what we call the zoonotic hazard.
Maria, sorry to interrupt you, but can you share your screen? Because I now hear that it was visible here, but these are apparently your own screen slides.
Oh, so it's my slides from me, right? Or you're showing them?
Yeah. So if you share your screen, then things become okay.
Okay. Is it showing now?
Yeah, almost. You put it in presenters mode, please.
I did.
Yeah. Now it's excellent.
There?
Excellent.
Oh,
yeah.
Okay. Sorry about that. Yeah, so as I mentioned, the zoonotic hazard is a measure of what we call measured as a density of infected ticks, so the number of ticks and their infection prevalence, and that has to be combined with a measure of human exposure to determine risk. Again, as Helen mentioned, the blacklegged ticks in the United States, and it's very similar cycle as Ixodes ricinus in Europe, is dependent on deer for reproduction. So deer are keystone hosts for the ticks. But they are not competent for Borrelia burgdorferi, which relies on a community of hosts to be transmitted. And these hosts vary in their competence. So white-footed mice, for example, in the US is the most competent host, as well as chipmunks as well. And so a more diverse host community has been proposed to dilute the infection by providing alternative meals to the ticks. So that's part of what I'll be discussing about this effect of biodiversity as well in my talk. So Lyme disease continues to emerge and expand geographically in the United States, as shown here. We have an estimated 300,000 or more than 300,000 cases of Lyme disease a year in the United States. And there's an estimate about 15 tick-borne diseases in the US, combining 3 different tick species. So it is definitely a very important disease risk, and also it's increasing in prevalence, and also there's new tick-borne pathogens have been discovered in recent years. So tick-borne diseases as well are emerging in urban areas. In New York City, for example, there's shown here, cases continue to increase. And although many of these cases are actually from travel, people going to other places outside the city, we estimated that 53% of cases have no travel history, in particular in the borough of Staten Island in New York City, which is the area that we're focusing our studies on. So there's definitely getting Lyme disease in the city. In Pittsburgh, for example, there's another study showing in the yellow line here how there's an increase in Lyme disease over time. And that increase is linked to an increase in cases in more urban zip codes and a decrease in more rural zip codes. So there's a shift in Lyme disease to being more urban than rural. In Boston, for example, there's also ticks. This is a press conference, but there's not a lot of published studies in the US on, or I would say none, or just, very, very few studies on urban tick-borne diseases in the US. So we know blacklegged ticks are strongly associated with deciduous forests. As again, Helen mentioned, this is a study that's about to come out from our group. It's a review showing that deciduous forests, leaf litter, canopy, and shrubs are really linked to tick-borne, blacklegged ticks abundance. And pine forests and grasslands are not associated with tick-borne ticks, blacklegged ticks in particular. So given that that forests are so important, there's been a proposition that reforestation that occurred in the last 150 or so years in the United States, in the Northeast in particular, and the Midwest, is the culprit, basically, has been driving this Lyme disease epidemic. So we saw an emergence of deer in the last century and followed by Lyme disease more recently in the 1970s and 80s up to today. So this, of course, is a correlation, but there is a consistency in the sense that forests are needed for deer and ticks, and those are the determinants of Lyme disease. However, a somewhat contrasting view is that it's not more forest what's causing the Lyme disease epidemic, but it's the fragmentation of the forest that is driving the epidemic. And this is related to the idea of the dilution effect. So forest fragmentation would have two main effects in terms of risk of Lyme disease. One of them is affecting the zoonotic hazard. So the concept is that larger forest patches will have a more biodiverse community. As they get fragmented, they would increase the amount of edge, and that's a habitat that's preferred by mice and other hosts of ticks and Borrelia. And the smaller patches would also release a predator mice, for example, from predator and prey competition, predation and competition. So this would lead to patches that are less biodiverse and more dominated by mice, which are highly competent, and there will be more risk. Now, fragmentation would also, independently of producing this effect on zoonotic risk, would expose people more to ticks. Just the fact that people are next to forested areas will intrinsically really increase their risk of tick-borne diseases. So these are two different effects, simultaneous, but they're hard to separate. But they are having pretty different effects. So to summarize some of this view of more forest, more Lyme, or less forest, or fragmented forest, less Lyme, there was a review in 2013 that tried to summarize this. So in the x-axis, there's a range of forestation going from a city with not a lot of forest to a very diverse large forest. And so initially, there has to be a positive effect because the more vegetation in an area with no forest at all will increase the hosts for deer and mice and other hosts for tick-borne diseases. However, at the very large level of forestation, we might start seeing this dilution effect, which is, again, still quite debated, but that is definitely a possibility. So in urban areas, of course, we are to the left of this graph. And something we can think about is how the cities are designed in terms of this fragmentation effect. So if we had a fixed amount of forest and we want to distribute it across the city, there's two kind of extreme ways in which we can do that. One has been called a land sharing design, where we have small patches of forest throughout the city. So sharing, because a lot of people can access those forests, but they're very small, so they can only accommodate a certain amount of biodiversity potentially. Or have them together in a sparing design or land sparing, where we have big patches of forest. And then some people will live far from it and not have access to them, but then we can accommodate potentially more biodiversity in a large patch. So according to the dilution effect, the zoonotic hazard will increase in these small patches, and also people exposure will increase compared to the land sparing design. But this all depends on how connected these patches are. It's in particular for deer. So this functional connectivity is really important, and how connected they are to people, how people are using these urban spaces. So to summarize all these ideas, we put together we thought of Lyme disease as a coupled natural human system. This is an NSF grant that we're currently funded for. And the idea is that we would model with data that I'll show you in a minute, the zoonotic hazard and the spillover effect on people living next to these forests, and that would correspond to the zoonotic hazard. And so ticks from the system can bite people. People will have adaptive behavior, so they can adapt their activities or protect themselves against ticks. And people can also have mitigating behavior, where they will reduce that risk by having interventions at the household level or more at the community level. And of course, this individual risk will be translated into community risk and Lyme disease incidence. So with this framework in mind, we started designing these agent-based models where we can simulate different types of landscapes shown here, where there's zoonotic hazard that can spill over into people's homes. And we also simulate movement of hosts as well as of people. And we can incorporate rules of movement to inform some of these models and different behaviors. So basically, we ask the question with the system, how does landscape composition and configuration affect risk? How does the connectivity, the functional connectivity through the mobility of hosts and humans affect risk? And then we can simulate how different human behaviors also can affect risk. So I'll talk about these 4 things in order. And first, I'd like to introduce Staten Island, which is the borough of parks. So Meredith Van Acker in my lab did a study throughout New York City surveying ticks. And she found that most of the parks are too isolated. Even if they might have suitable vegetation, deer just can't get there to have established population of ticks. So on Staten Island, where the parks are very connected, that's where she found the most ticks and established population of ticks and infection. The forests look like this on Staten Island. They're really mature forests. So the field data we are collecting to estimate the zoonotic hazard, we're collecting camera traps, trapping mice, collecting ticks. And we are putting GPS on deer and raccoons in particular, which are hedgehogs, I guess. So they move around the city a lot and carry ticks to estimate the functional connectivity. Then the human connectivity we estimate with the Tick, an app, a smartphone app, where we assess movement and behaviors. And then we look at spillover by sampling backyards of people that live next to those houses. Those are the only ones that can really get ticks from the hosts that come from the parks. And we do similar assessments as well as surveys to assess people's behaviors. And then we also do tick assessments in the parks. So the first thing from Meredith's study, we found that the vegetation surrounding the parks is a very important determinant of the density of infected ticks. So she found that the percent of trees was positive around the parks was positively associated with the density of ticks, and water and soil around the parks was negatively associated presumably because it reduces the connectivity. So I'm going to skip this, I think.
So I ask you to wrap up the presentation? I'm really sorry, but 12 minutes is of course short. But
okay, so we assess the length. Yeah. 1 minute. So we assess the landscape connectivity across the parks was the most important determinant of infection. And we are assessing connectivity of humans, shown here, and sorry, I don't have time. And the other thing we looked at is human movement, and we saw that they're shown by using the Tick app data, that there was a shift during the COVID epidemic from from use of the parks to use of the backyards. And there was a 30% increase in ticks in that situation. So yeah, so I'm going to have to cut all this. And there's we could simulate basically with these models adaptive behavior and people avoidance of parks, shown here, and assess how they reduce their risk. And I'd like to just wrap up. I will have to wrap up the presentation. So just to take home, the intermediate levels of forestation and biodiversity increase the risk of tick-borne diseases, and smaller urban spaces could increase the number of mice that are most competent hosts, but the functional connectivity is what determines whether the possibility that the ticks will persist and Lyme disease will be transmitted. People's behavior are really key, of course, for exposure, and we need really to couple natural human systems to understand any interactions and feedbacks between the natural and the human system that may affect risk. So I'd like to thank all these people, and sorry if I went over.
Yeah, thanks a lot for the presentation, Maria. And I know the feeling, because it's always you're really full of your work and you want to share it all with us. So it's the best intentions. So thanks a lot for this presentation. And what I learned is that we actually can see a city like a fragmented forest, which is, I think, a very nice perspective. And well, if I can ask one question. If you have this, how can the increase in biodiversity help kind of reduce the burden of mice or deer in systems like this, if you have this fragmented forest?
Sorry, can you repeat the question?
So the question is there a way we can increase biodiversity in fragmented forests to reduce the burden of mice and thus ticks?
I think the difficulty is, well, first, the biodiversity is already relatively low in urban areas. And I think the problem with thinking of biodiversity is when we say biodiversity, which hosts are you increasing? Which I think it's a problem with tick-borne diseases, because most of the hosts of tick-borne diseases are very human adapted. So we're not talking about hosts from pristine areas. So, for example, raccoons. So if we say we increase, raccoons are biodiversity, and they would reduce potentially the infection, but they can also carry a lot of ticks. So I think we should be thinking about which hosts might be having different functions rather than talking as biodiversity as just a general term, especially in these situations where hosts have very different behaviors and abundances in humans and responses to human resources. So some, as Helen said, foxes or raccoons may be eating trash, are trash. So then they may become extremely abundant. So the dynamics are very different in urban areas. So I think we really need to revisit a little bit of the concept of biodiversity as a one thing, as a one number, let's say.
Yeah. Thanks a lot for this answer. Thanks a lot for your presentation. It's really much appreciated by our audience. And I would now like to go to our final speaker of this session, who is also from the US, Mary Gardner, who is a professor at Ohio State University. She is interested in ecology of urban green spaces. And the title of her talk is Reimagining Vacant Land as a Resource for Urban Bee Conservation. Please, Mary, the next 12 minutes are yours. And audience, please keep sending your questions. I think we have a bit difficulty in finding the connection, although I think we've seen her online.
Great. Good morning. Sorry, I was just looking for the presentation. So can you see my presentation now?
I can see your presentation, yeah, and I can also hear you.
Okay, great. Well, welcome everyone. I'm going to be talking to you about vacant lots, which are a issue, but also opportunity in shrinking cities around the world and their potential value for urban bee conservation. So shrinking cities are novel environments where we have lost large portions of the population of people. So there are about 450 cities worldwide that have experienced this substantial population decline. And in the United States, many of these cities are concentrated sort of in the middle of the country in the Midwestern region, and they are historically areas where manufacturing activities occurred. And those areas are shown in red in this map. I am in the state of Ohio, and we have several shrinking cities, including Cleveland, Ohio, shown here adjacent to Lake Erie. Cleveland, Ohio was once the fifth largest city in the United States, but it has lost 50% of its peak population. And when 50% of the people move out, you have an overabundance of infrastructure that is no longer needed. So as you drive around in Cleveland, in the inner city neighborhoods, you see many abandoned homes, which are boarded up and going into disrepair. Every summer, when the city is able to, they remove about 1,000 structures per year. There are demo plans for the next decade of homes yet to come down. So the amount of vacant lots in Cleveland is expected to continue to grow. What's left after these structures are removed are weedy green spaces of early successional vegetation, such as shown here. This is red and white clover, Queen Anne's lace, and chicory, which are common early successional weeds in our area. These habitats are mown once per month by the city, and so in between those mowings, you have this early successional vegetation. Cleveland has a lot of vacant land. As I said, they lost half their population. The city manages 30,000 vacant lots. This map shows the city outline in gray. All of the green areas are existing parks. The red areas are vacant lots. You can see that there's a substantial amount of them. These areas are considered permanently vacated. It's unlikely that anyone will move back into these areas. And so this represents a major shift in green space. So I was very interested to know whether these newly available green spaces would be valuable bee habitat. And so one of the first things we did was just simple pan trapping, where you put out little pans of water, and we did observations within vacant lots. And through these activities, we found 107 species of bees, and that's about 20% of our state's fauna, which really was impressive to me. Even more exciting, the majority of the species were native, and many, depending on the type of area that we sampled, whether it was a very complex landscape or a very isolated patch, were ground nesting. And historically, ground nesting bees are thought to do really bad in cities because, of course, there's not a lot of habitat for them to nest. But when we added all of this green space back in, there were more species able to exist. And so this led me to want to see, well, are there things we could do to make vacant land even better for bees? And with funding from the National Science Foundation several years ago, I set up a large citywide research project to determine if different types of plantings that we might establish on vacant lots might improve the habitat quality for bees. And these types of habitats ranged from this T1 picture in the corner is our city managed vacant lot, so mown once a month. And then we had all kinds of different lawn treatments, as well as native, what we called pocket prairies, which were plantings of Ohio native plants. And in this short presentation, I'm just going to focus on some key results that we found comparing this traditionally managed vacant lot with these pocket prairie sites that we established. So here, just to show you, is a close-up of a control lot. So there's the early successional weedy veg. And then here on the right is a pocket prairie site. These were tall prairie vegetation, so native plants known from Ohio prairie sites, and also weedy plants would work their way in. So it was a mix of different plant species. And here are some pictures of our pocket prairies throughout the summer. They're really beautiful sites, but very challenging to manage. And I'm happy to talk to folks about that if you want to contact me. So what did we find? Well, our first question was, does creating pocket prairies enhance bee species richness? And interestingly, we found that the same number of bee species foraged on these early successional weedy plants as they did in the pocket prairies. What mattered more to the bees is that there were a lot of flowers. So as bloom abundance in a site increased, we had more bees, but we didn't see a difference in richness when we planted the pocket prairies. I'll note I'll be going through just some major findings here. If you want more details, all the papers coming out of my lab are shown in green here, and I'm happy to send those. I can put my email in the chat. And while this might seem disappointing to some, I actually think this is really a positive because it means that anyone can engage in creating bee habitat that's beneficial. We can't plant pocket prairies everywhere, and so the weedy vegetation that is present is also valuable. But just because you see something out there foraging does not mean that it has a high quality of health. And so we also wanted to know, does creating pocket prairies improve the quality of the habitat for bees? And to look at habitat quality, we focused on reproductive success. So we put out these little stem nests, the bees, cavity nesting bees nest in here, and we then x-rayed these nests and we could see how many larvae are in here. So each, what you're seeing in this straw are pollen balls deposited by a female bee, and she's laid offspring in each one of these cells, and the larvae get bigger as they eat the pollen ball. So here with the biggest larva, it has the smallest pollen ball. So we x-rayed these and counted the number of larvae. And what we found was the number of native larvae produced in a site actually did increase with the flowering prairie compared to the control lot. So establishing our pocket prairies did improve bee reproduction compared to the control lot. We also wondered, does it matter where you put these pocket prairies? So in some cases, landscapes have neighborhoods that are pretty well intact with maybe just one isolated vacant lot. In other parts of the city, you have huge areas of vacancy. So we conducted this landscape analysis where we pulled maps surrounding each one of our vacant lots, and in a 1,500 meter radius, we determined, well, how much green space is there and how much impervious surface and buildings are there surrounding each site? And does the connectivity of the surrounding green space influence the success of the pocket prairie as a bee habitat? And what we found was the connectivity of green space was very important, both for bee communities in those traditional vacant lots and in our pocket prairies. We found that when there was more habitat connectivity, we had a higher species richness, more small bees, which might be challenged by their dispersal capacity, and a greater reproductive success of bees occurred in sites with lots of green space surrounding them. Now, when doing these habitat alterations, we also have to think about the history of the city. This was an industrial city. They produced iron and steel. It's now post-industrial. What happens when you produce those metals? You get heavy metal contamination in your soil. When we worked with community scientists to sample bumblebees out in the city of Cleveland, they contained heavy metal contamination that we could detect. So the big question was, well, are we creating conservation habitats in a place that then has negative consequences for bee health? So in a field experiment where we set up tents, we actually took the levels of heavy metals that we found in bee provisions in Cleveland, and we fed that contamination to bees that we could control their movement in these field tents. And unfortunately, we did see a negative consequence on health due to exposure to heavy metals that we find in the city. So here we have hives that were just fed sugar water. They had some mortality, about 20% brood mortality. But those fed heavy metals had 3 to 4 times the brood mortality. And when they received this toxic cocktail of all heavy metals, which is pretty relevant because it's very realistic based on the conditions we found in the field, they had about 9 times the concentrations. So most of my work right now is actually looking at how bees come into contact with these metals. Do the plants take it up, or is it due to dust deposition on the plants? And how do we mitigate this? There are many different methods that you can use to reduce the bioavailability of heavy metals. Finally, when you're working in a community, many people think, well, what do you do in terms of the neighbors? Like, what do the neighbors think of these scientists out there creating these pocket prairies? Do they like it? Do they hate it? Well, I'll say we were not uniformly loved or hated by the community, but we did work with the community quite a bit to determine what sorts of habitats might be best suited for an urban environment. When you talk to folks, many people do not like tall vegetation. So this is a news story. Cut it. Residents on the east side say Cleveland needs to mow the grass. So when the city doesn't get out early in the season to cut the grass, people get upset. So this is not meeting local landscaping norms. People expect these sites to look more like mowed turf. Also, people can be afraid of bees, right? So not all neighbors think pulling in all of these bees is a good idea. There's also safety concerns. These are neighborhoods with very high crime rates, lots of gun violence, lots of drug activity. And so we can have major challenges in creating sites like pocket prairies. So for instance, this picture here, picture A, is a pocket prairie near a bus stop. Picture B is my student, Denisha Parker, and I, when we held an event for kids in that site, collecting bugs. It was a very positive event, but not long after that, a young person was murdered right around the corner. And we then cut this site because a neighbor was very worried someone might be hiding in the plants. Now, these are not very tall plants, but it doesn't really matter. If someone is worried that there could be someone in there ready to cause harm, that's not going to balance out the benefits that that site could have. So these are really challenging issues that I'm not able to get into in depth here, but just want people to realize the complexity in creating these habitats.
Can you please wrap up your presentation, Mary?
I'm going to. So are vacant lots valuable bee habitat? Well, they certainly can be if we're able to balance the dispersal needs, foraging, and nesting needs of bees with the aesthetics, safety, and values of the people that live near these sites. And I would suggest heavily to people to start by co-creating these habitats with the community and then considering some of the design, landscape, and pollution challenges that I've presented today. Thank you very much. I would like to acknowledge our funding and partners, and I'm happy to take any questions. And I've added my email here, and I realize in haste made a mistake. It's Gardner.29, not Gardner.20. And so if anyone would like copies of those present publications, I am happy to send them. Thank you so much for including me.
Thank you so much for your presentation. Much appreciated. And I'm sure if people make mistakes in the email, then we can help. Could you please turn on your camera? Because then the audience can also see you yourself.
Oh, I'm sorry.
Because I think you deserve it with a presentation like that. So thank you for that. a question. So did you also consider other insects like as bees, for example? People like butterflies a lot. Did you also look at other insects?
So weirdly, we did not look at butterflies, even though they are beloved by many people, but I have looked at many things. Other insects, we have studied extensively ground beetle populations, spider communities, lady beetle communities, and also the value of the urban forest that occurs on vacant lots. So those are also research topics that I work on.
Okay, thanks a lot. Well, another question. So you also find non-native bees. Do you have any idea where they come from?
So we found a total of 12 species of non-native bees, one being the European honeybee. So that's obviously, a bee people cultivate. We did find 11 types of non-native bees. Some of them have been in the United States for a long time. And so things like the wool carder bee are very common here. We would find that. We did actually have a new North American record of a bee species in Cleveland in our data, which was really surprising. And, how these species get here is kind of a mixed bag. Probably if they're a soil nesting or cavity nesting species, they're probably coming in as cocoons in plant material or wood products, things like that, and then released into the environment. So cities are often a place, as many of you probably know, where we look for invasive species. But I was actually really surprised at what a low proportion invasive species made up of the total bee community that we found.
Thanks a lot. Yeah, it's also the experience here that nowadays cities are often more biodiverse than the rural areas, which is, striking in itself also for the other part, of course. Again, many thanks for your presentation. It's really much appreciated. Well, this concludes the first part of this webinar of the Netherlands Center of One Health on green cities. But please do not go away, because we have again for you an energizer like in the beginning of this webinar for 3 minutes. And after that, you will have a short break in order to do, things that cannot wait anymore. But please be back here at 5 minutes past 4 Central European time. I say that for the foreign guests. Please be present back at 5 minutes past the hour, because then another exciting part of this webinar will take place. You will see 10 pitches of PhD students from the Netherlands Center of One Health, so a wide diversity of topics. And also in that part, your participation is really very, very important, because at the end, Maarten Hoek, one of the organizers, will show you a Mentimeter poll where you can vote for your favorite pitch. And that person will obviously win the awards that we will give away here at the end of the webinar. So please be back here at 5 minutes past the hour.
Oh, I feel so well educated after hearing out the three ladies. And so sad, because I think there is no other option than to move. But where to? I don't know. Since an hour I am down, Amsterdam is my hometown. All my life I felt so swell, looking over the canal. But from what I understood, life here is no longer good. I was fine between the bricks, and I had my daily kicks, going to the opera house or museum with my spouse. Oh, so much worth being seen, but my city's getting green. Lessons learned here from three chicks. There are risks involved with ticks. They warned us in every way that these ticks are here to stay. The conclusion of their thesis, ticks spread terrible diseases. Helen took us by the hands, talked about the Netherlands. Greening means some problems solved, but also new risks involved. For everyone who wants to join, there are two sides of the coin. Green cities. Green cities, green city, it ain't pretty. It's Maria's expertise, emerge of tick-borne disease. Humans and nature interact, and the health risks are a fact. Where green spaces are connected, cities cool but get infected. Mary gave us little hope, Vacant land across the globe, pocket prairies for the bees, so she told from overseas, followed by a sad conclusion, bees are victims of pollution. It ain't a love marriage between human beings and the green. It's a tense relationship, you can easily lose your grip. But these three ladies I heard talk, they look solid as a rock. I suppose they stay away from the grass, the trees, the hay, avoid stone, martyr and deer. When they see hosts they disappear, and they don't drink and they don't smoke, and they never plant an oak. Green city, green city, green, green city, it ain't pretty. It was clear when I moved here, reasons enough to live in fear. Every corner saw a threat, I took risks without regret, took my bike and went ahead, even when the lights were red. There are junkies everywhere, tokers at Museum Square, and when spring is in the air, women in their underwear, and at night when it is dark, creeps across the Vondelpark. Helen made it very clear, there really is a lot to fear, and Maria made me see, there is no escape for me. Mary didn't turn me down, but it's a jungle in my town. Psychopaths and terrorists are no longer on blacklists. Oh, the lessons learned today from what these ladies had to say. The biggest threat right from the start seems to be my own backyard. Green city, green city, green, green city, it ain't pretty. And again, green city, everybody. Green city, where are the little hands? Green, green city, it ain't pretty. Ooh! Thank you.
second part of this webinar of the Netherlands Center of One Health. We of course like to thank Dominique for her excellent contributions in the energizers during the break, but also of course the song she made also with the ukulele before the start of this webinar. In the second part you will see 10 pitches of PhD students who are affiliated with the Netherlands Center of One Health, and they will pitch their work in 3 minutes, and then, well, they will do that subsequently. But please pay attention, because at the end you can vote for your favorite PhD student, and the one with the most votes will win this contest. And the first one I would like to introduce is Ayla Hesp. And Ayla is working with Wageningen University and Research Center. And the title of her talk is The Use of Whole Genome Sequencing to Monitor Antimicrobial Resistance in Livestock. Ayla, please.
Thank you. Hello, so nice that you are watching. I would like to share with you my research on the monitoring of antimicrobial resistance, AMR. And what I have for you today is a puzzle. You can see it on the slide. It's a multicolored puzzle, and you can see the colored pieces of information, but it's hard to see how these fit into the bigger picture. And that is also how it is with AMR monitoring. It's a challenge to translate the data into useful output for monitoring activities. So what I show here is a phylogenetic tree of E. coli strains sampled from animal species, 3 different types, and they are indicated in colors in the first row of the tree. You do not have to see all the details, but in the enlarged part, you can see that the first row is yellow because it's all broilers, or orange yellow. And the second bar indicates which farms these animals were sampled. And it's the same farm in this case, but because it's the same shade. So these are 3 isolates which are from the same farm. And what we did was add the metadata here of the resistance genes and plasmids that were found in these isolates. And these are indicated in the blue circles and in the green circles. And what's the nice thing of whole genome sequencing is that you get the whole genome of the bacteria. So we do not only know the resistance types, so the antimicrobials where these isolates are resistant for, as we previously had, but we also now know whether these bacteria are related and which other genetic information they carry. And this example in the red box shows 3 genetically identical clones that's present or visible from the tree, and they carry the same resistance and also the same plasmid replicon types. Now, these plasmids are mobile genetic elements which allow the resistance to spread between bacteria. So this is just an example, but this is to illustrate what this whole genome sequencing method can be of added value for AMR monitoring purposes. If you want to know more, I would invite you to read my PhD thesis, which is hopefully finished very soon, because this is also for me the last piece of the puzzle. Thank you.
Thank you, Ayla, for this presentation, and I think it's really good to see that this genetics enter in the antimicrobial resistance era on top of the phenotypic evaluation. Can you give us some guidance on what would be best sampling strategies to see this difference between clonal spread and other types? Because of course, often you cannot test all animals on the farm. Do you have some guidance on that?
Yeah, it's a very interesting question, because in regular monitoring activities, we normally take only one animal we sample one animal per farm. And we do that actually to avoid clustering of the outcome data, because if we would take always multiple samples, we would probably get a sort of bias in the outcome of the resistance genes monitoring. So for regular AMR monitoring for detecting trends, which is the main goal of what we usually do, it's best to just take only one animal from a farm. But if you want to determine specifically in-farm prevalence, it's better to take more, I would say at least 10 animals, but more animals from the farms. But specifically for AMR monitoring, it's not the best idea.
Thanks a lot, and your presentation is much appreciated, as is your answer. But now I have to let you go because we need to continue with the show. The next candidate who will now enter just after Ayla has left will be Remy Muts, and Remy is working at the Utrecht Medical Center, and the title of his presentation is Understanding Antibodies and Their Potential to Combat Antibiotic Resistance. And you have seen the challenge. All these PhD students are challenged to do it with one slide only. Please, Remy.
Yes, thank you. So several years ago, the World Health Organization estimated that by 2050, over 10 million people a year will die due to antimicrobial resistance. On top of that, the current situation our world is in now only enhances this problem of antimicrobial resistance, as many people end up in hospitals and get and receive many antibiotics as a prophylaxis. This is also dubbed the silent pandemic. Especially for one type of bacteria, the gram-negatives, we are running out of treatment options, as illustrated by the fact that the last class of antibiotics to treat these gram-negative bacteria was developed over 50 years ago. Luckily, it's not all bad news. We all have a great immune system with several ways to combat these gram-negative bacteria. A mechanism from this, which we've all heard of, are antibodies. And antibodies, in turn, have several mechanisms to combat these bacteria. One of them is by activating the complement system. The complement system, in turn, has also several functions, but for now I will just mention one. That is, that in case for gram-negative bacteria, it can form holes in these bacteria which directly kill them, which you can also see on the slide here with the bacteria depicted in orange. That antibodies can activate the complement system, which induces the killing of these bacteria, has great potential. It means that with the right antibodies, we have a new therapeutic strategy to treat antimicrobial resistant bacterial infections. And this strategy has a big advantage because it uses our own body, our own immune system. However, in order to use this beautiful strategy, we do need to know how this works. And that is where the gap in our knowledge lays. We do not know yet why one antibody does this better than the other. And this is also the aim of our research. We are investigating why some antibodies are way better at activating the complement system and therefore in inducing the killing of bacteria, and why some other antibodies cannot do this. So with that, our aim is to investigate antibody-induced complement-mediated killing of gram-negative bacteria on a molecular level as a basis for the development of a new therapeutic strategy to tackle antimicrobial resistance. With that, thank you, and I'm happy to take some questions.
Thanks a lot. Yeah, unfortunately, we only have time for one question. Because we need to be quick. Well, interesting new strategy. Is this just a new strategy, or does it also have more potential towards that it's more difficult to evoke resistance against this new strategy than towards the regular antimicrobials?
That's a good question. It's difficult to say now because it's not really in use yet. So you can only say that once you're trying it out. But as the response is more broad, so it's several functions, and except direct killing, you also stimulate your own immune system in other ways. So it is less likely that it will develop resistance than the use of current antibiotics. Okay, that makes it even more interesting. Remy, thanks a lot for your presentation. And now I also need to ask you to leave the room and make a place for the next one. Thanks a lot. Thank you. The next presenter will be that will enter the room now is Juriaan van Eersel. And Juriaan is affiliated with the Royal Netherlands Academy of Sciences. And his presentation is entitled Birds as Vector for Infectious Diseases Along the Rural-Urban Gradient. Juriaan, please.
Yes. So hello, my name is Juriaan van Eersel and I'm a PhD student at the Dutch Center for Avian Migration and Demography at the NEO-KNW in Wageningen. And my PhD project is part of the One Health Pact, which is a research collaboration of 26 PhD students and one postdoc student, a postdoctoral researcher, where we together take a proactive approach in preparation of future mosquito-borne disease outbreaks. And birds play a very important role in the transmission cycle of mosquito-borne diseases, where birds are, for many of these diseases, the main host, where they act as an introduction and amplifying host species. And some of these pathogens have the ability to also spill over to humans. And within birds, for a particular life stage, we think that young birds, those that fledge the nest, are very interesting and important in amplification effects. So what we see is that these young birds are still naive individuals that have not been exposed to many pathogens and therefore may have a higher risk of infection. And also, the mosquito abundances for these mosquito-borne diseases are very close to the period when many of the fledglings have hatched, or has fledged the nest, increasing the risk of those little birds to get infected and reach high viremia levels to subsequently infect new mosquito species. So to investigate this really further, we started this year with coloring birds in the nest, searching and monitoring blackbird nest, and coloring the birds in the nest, but also take a blood sample. And these will be investigated on active Usutu or West Nile virus infection, and also investigate their immune system. And the interesting thing with coloring the birds is that we can subsequently recatch birds within the same year or in the next years to investigate the development of the immune system over time. Yeah, sorry. Oh, sorry. Yeah, I was a little bit distracted. So that means that we can recapture those individuals over time. And we predict that with urbanization and urban greening, that there will be a lower immune or weaker immune system of those nestlings and fledglings compared to those in more natural sites. And that was it.
Okay, thank you. And then the obvious question is, why do you think that there will be a lower immune system in these fragmented forests, as we've heard them in the previous talks?
Yeah, and yeah, that's an interesting thing. So the first hypothesis, of course, that there's a limited food conditions in urban habitats because there is there's a lesser choice of insect species, for instance, or earthworms. But on the other hand, it could also be that the earthworms and the insects that they get have, for instance, heavy metals inside them, which also have a direct negative effect on the immune system of the animals.
Okay, so interesting. You study Usutu and also West Nile. Now, as from last year, we do have West Nile virus in the Netherlands. Is that going to be so the greening of cities? Is that going to be a disease of concern in the coming decades?
Well, yeah, for Usutu, we don't really know. Usutu for infections seem to be declining in birds. But West Nile infections, for instance, we had last year the first West Nile case in the Netherlands in the common white throat, which was shortly followed by a range of human infections with West Nile virus as well in Utrecht and in Arnhem, which of course increased concerns that perhaps this year we have another influx of West Nile infections in both birds, but also in humans.
Yeah. You did a very quick presentation, so I can still ask another question that interests me. So this recapture of the birds, can you give me any indication on which proportion do you actually recapture in the city? So do these birds stay in the city just because they love their site, or are they also migrating over larger distances? Yeah,
that's also a tricky question because honestly, we don't really know. We don't really know where blackbirds are moving and where they are going and whether they stay in certain areas. So will be also the next step for next year, that we will also use GPSs on birds to track directly within a year where they are going exactly. But for now, within a month after they fledge the nest, we can recapture them, and we already did recapture some of the nests. So we have samples from them being a nesting, and then subsequently a month later as well. So we can already compare the immune status and the immune differences, but also the infection of Usutu or West Nile.
Very interesting. I'm sure we'll see you back here, but now I have to let you go because we need to continue with the pitching show. Thanks a lot, Jurjen.
You're very welcome.
And so that leaves me to announce our 5th PhD student, who's entering just now, who is Jo-Anne Verschoor, and she's affiliated with Leiden University, and the title of her talk is The Search for Novel Plastic Degrading Enzymes. Please.
Yes, thank you. I would like to take you all with me on my road to the station. It's a 20-minute bike ride, and on this bike ride I encounter several places that look like the one you see on this slide. The amount of plastic waste that I encounter on a daily basis amazes me every day. And I always wonder how it can be that these amazing materials with their great features are being discarded after such a short lifespan. The answer is relatively simple. Plastics lose their value after use. And this is because the recycling of plastic is a very tedious and expensive process, which leads to a very low quality product. So, that means that manufacturing new plastics is simply more expensive and feasible than gathering and recycling the old. So what we need to do to overcome this problem is that we need to increase the value of these plastics. And we can do that by developing a new recycling approach that leads to a high quality product. A very good and promising approach is enzymatic degradation. Enzymatic degradation focuses on the degradation of these synthetic polymers into their original building blocks using enzymes. These original building blocks can then be purified and used to manufacture new products and new materials without the loss of quality. That's the most important thing. But the main question is, where do we find these enzymes? Well, the answer is actually already in the picture that you see on the slide. Because in these polluted areas, some microbes have already developed the ability to degrade these plastics. It's just that their enzymes are not efficient and abundant enough to do so. So what I do in my research is I isolate these bacteria, I identify the enzymes that are responsible for the degradation of these plastics, and I optimize them and produce them in high quantities. The bulk production of these enzymes is the key to boost enzymatic degradation as our main recycling approach. But we're still at the start of this race, and I really hope that in 1, maybe 2 decades, these pictures will be pictures of the past, and we can all simply ride our bikes in our green cities, that I hope we have then.
Yeah, we all hope that, because it's a much nicer view than seeing these pictures, I think, which we all recognize. So very interesting. So we see in the society that there are a number of different approaches regarding the plastic problem. So one of course is also, well, making things, at least returning some money on PET bottles. Or do you think that this is the solution, or is it part of a solution? Which solution set do you see for the problem?
I think it's a part of the solution, because what we see now is because these plastics and other waste have so low value, people throw it away. So what we see with the PET bottles is that because we pay statiegeld on them, you return them because you get money back. Yeah. But if we have such a system and you combine that with these sustainable recycling methods, we can have a very sustainable way of plastic recycling in the future. We're already heading there, because at the moment we're already getting statiegeld on the smaller bottles and on the cans, but we're still not there yet.
And then, well, another problem regarding plastic, which has gained quite a bit of attention in the past years, are these microplastics, which are well, apparently everywhere. Even if you look at the North Pole or in somewhere way deep in the ocean, you will find them. Could these enzymes or these bacteria also be a problem for this well, really widespread environmental problem?
That's always the question, of course, and that really depends on how the legislation is going to be in that time. Because there are already bacteria and fungi that we isolate from the soil that we see have these abilities. Not for all plastics, but for some of them. And they're already there. So it might be that in a very long time span, they will disappear. But still, it's such a tedious molecule, such a recalcitrant molecule, that it's hard to break for them. So either we have to gather it and we have to expose them to it, like on purpose, so then they can degrade it. Or we have to find a way to spread these bacteria. But then you have to do it via the natural ways. So isolating them and cultivating them and spreading them over soil, which is sometimes used in agriculture already.
Okay. Thanks a lot for this very interesting presentation. Johan, it's really much appreciated and for answering the questions. But I hope you also get photos like the others. And please, I have to, also to ask you to go now and make place for the next contestant,
No problem.
who will be entering now. And our next presenter is Michelle Molendijk, who is affiliated with the Erasmus Medical Center. And we are getting in a topic that we also previously had, Staph aureus, but this time it's phage therapy against Staphylococcus aureus infections. Please.
Yes, thank you. So as you heard from other PhD students, antibiotic resistance is a very big problem currently. So it's estimated by the WHO that by 2050, many people are going to die because they have infections that cannot be treated. One of the bacteria, as you mentioned, Staphylococcus aureus, is one of the bacteria that can be very antibiotic resistant, and that's also the focus of my project. So we need new solutions for antibiotics. And for my project, we are thinking about bacteriophages. So what are bacteriophages? They are viruses that can only infect bacteria, and they kind of look like this in the black and white picture. So they have this head where they store their genomic material. They have a neck or a tail and kind of feet where they bind to the bacteria. And phages are the most abundant entities on the world. So it's estimated that at any time, there are around 10 to the 32 bacteriophages on the planet. And they're everywhere. They are in water, they are in soil, they are in us. Next slide. I cheated with the animations. Yeah. So in my project, I'm looking at the interactions between bacteriophages and bacteria, of course, but also between bacteriophages and the immune system, because if you want to use them as a therapy, you have the immune system to take into account. So it's kind of a love triangle I'm looking at. So I'm looking if the phages can kill the bacteria, if the bacteria can get phage resistant, if the phages are still working, if bacteria form biofilms, but also are the phages cleared by the immune system, or can they work together to clear the bacterial infection? And in the ideal future, we have the following scenario where we have a patient coming into the hospital with an infection that cannot be treated with antibiotics. And you have all these phages on the shelf. You identify the bacterium and the location of the infection, and also take into account the immune status of the patient. So are they immune compromised, or do they have a healthy immune system? And also, is there biofilms, and which phages can we use to treat this infection? Of course, we don't know if this is possible yet, so it's up to me to figure this out during my PhD. Hopefully I can manage. If you want to contact me, my email is in this slide, and that was it.
Thanks a lot for this presentation, Michelle. I'm not a specialist in phage therapy, but I think some time ago I saw a documentary in which, there it seemed to be quite an old therapy. So it was apparently some scientific institute in Georgia, I remember, if I remember well, who were already working with it for a very long time. Why hasn't it taken off earlier?
Yeah, so actually bacteriophages are first discovered in 1917, I guess. Yeah, by a French scientist. But actually, that was around, I think, 5 or 10 years later, antibiotics were discovered, and antibiotics were cheaper and easier to manufacture. So in the western part of the world, we continued with antibiotics, while in Eastern Europe, they kept going on with phages. So they are more advanced in that respect, but they didn't really have solid trials, for example, to prove that they really work. So that's the Western world is now catching up to them, actually, to see if it's really something we can use. Yeah,
interesting. And can you explain me a little bit about the specificity of these phages? Are they really very specific for specific bacteria, and does that hamper their at least clinical application later on, or is it maybe an advantage?
Yes, so there are very specific bacteriophages that can even distinguish between bacterial strains of the same species. So for a therapy, of course, that would be nice because you don't mess up your microbiome, for example, which you do with antibiotics. Okay. But on the other hand, you also want phages that can have a broader spectrum, so you can get not only one strain of Staphylococcus aureus, for example, but multiple strains. So what we are looking for is kind of the golden middle road. So the phages that have a broad host range, but also do not interact with other species of bacteria.
Okay, thanks a lot for this presentation. I'm sure we'll hear more of you in the future.
I hope so. Yes.
But now I also have to ask you to leave and make place for the next presenter. Thanks a lot for your presentation. And the next presenter, number 7 on the list, is Esther Tax. And Esther is affiliated with the Radboud University Medical Center in Nijmegen. And the title of our presentation is, well, it was on the news, I think, some time ago, BCG, a vaccination in the elderly during the COVID-19 pandemic. Please, the floor is yours.
Yes, thank you for the introduction and allow me to pitch here. So today I'd like to present a study we've been doing in the past year on BCG vaccination in the elderly. As you're probably all aware, the elderly population has been hit hard by the COVID-19 pandemic. In the Netherlands alone, over 17,000 people, of which most were over the age of 60, have died in the past year due to COVID. And it's not just this outbreak that older people are more vulnerable to these infectious diseases. Due to their aging immune system, there are changes that result in them being a more at-risk population, and specifically the adaptive immune system, so the part of the immune system that is generally thought to be associated with a memory response, is most affected. Our research group is working on the innate immune system, which is the other part, that is generally thought to be more of a non-specific part of the immune system, and specifically trained immunity. And trained immunity is the functional reprogramming of innate immune cells, mostly, for example, monocytes, neutrophils, and NK cells, resulting in an improved response against microbes after another stimulation. So it could be a completely other pathogen. So with this COVID pandemic, no specific vaccine was available at the start, no specific medications were available. So we started thinking, can we use this method, this trained immunity, to give these elderly people some protection? One of the things that can train cells is the BCG vaccine. It's a vaccine that's been around for a while and is usually used for prevention of tuberculosis. But we know from some small trials that it can also, in the elderly people, show a reduction in respiratory tract infections. So in the first wave of COVID last year, April 2020, we started a new trial in 2,000 elderly people, the ages 60 and above, and our oldest participant was 93 years old, in which we gave them a placebo vaccine or the BCG vaccine. And over the past year, we've followed up with, they would register any health complaints they had, the severity of this, doctor's visits, COVID tests, and COVID contacts, to see whether there is a difference between these groups. And at the end, they also draw some blood to mainly check antibodies, but also in selection, some cytokine responses. So we're hoping soon we can share some interesting results and hoping to see whether this BCG vaccine can provide some general protection against infectious diseases. Thank you.
Thanks a lot for this presentation. Yeah, I also knew BCG in particular for tuberculosis vaccination. So given that we're now in the situation where, of course, we have specific vaccines against COVID-19 and almost in particular the elderly have all received these vaccines. In that situation, do you still see a role of this?
Well, I wish this would be the last pandemic we'll have, but we'll probably have some in the future, or other infectious disease outbreaks in which it takes, again, a while to get to a specific vaccine. Now it was a year until we got a specific vaccine, which is incredibly fast. So in case of a new pathogen emerging, it will be good to have an alternative it will not give specific protection, but could give a sort of boost to the immune system so that the most vulnerable populations are less at risk to get severely ill from these new pathogens.
Yeah, and would it in research also be possible to discover which part of this this bacillus would at least evoke that, or would we still need to use the vaccine as a whole also in the future?
Well, I know we're looking at several in vitro still at some other stimulants that induce this training capacity. And I know in Germany there's also a team working on a variation of the BCG vaccine that has specifically at the specific parts that might be able to induce this strained immunity. So it's not the entire Bacillus calmette-guerin, but like a part of it.
Yeah. Okay. Thanks a lot for this interesting talk and for your answers to the question. It's really much appreciated. And yeah, also now I have to ask you to leave the room again, like I did for all the others before, to make place for the next PhD student. So thanks a lot.
Yes, thank you.
And the next PhD student to present for you, to pitch for you, is Sam Noy, who's entering now. And he's affiliated with the University Medical Center of Leiden. And the title of his talk is Fecal Microbiota Transplantation Influences Procarcinogenic E. coli, I say, in multiple recurrent Clostridium difficile infected patients. So a combination of two bacteria. Please, the floor is yours, Sam.
Thank you. How do you prevent cancer with poop? I would like to introduce you to my research, which is about gut bacteria and feces or poop. And this work is a collaboration with the Netherlands Donor Feces Bank and Vedanta Biosciences, an American company. And yeah, as the title suggests it's a mouthful, but it's about fecal microbiota transplantation or poop transplantation as a therapy for curing diarrhea caused by multiple recurrent Clostridioides difficile infections. So that's still a mouthful. So it's about fecal microbiota transplantation, which has been proven to be a very effective therapy to cure this infection. And we think it works by taking the bacteria from the feces from a healthy donor. These are introduced into the patient to re-establish a healthy gut microbiota, so the bacteria that live in the gut, and then the patient often cures after that. But in recent years, there's been growing attention for the potential role that bacteria play in the development of cancer. Specifically, there's a variant of E. coli, so-called PKS E. coli, that creates a toxin that causes a specific mutation that has been found in patients with colorectal cancer. And this same E. coli also occurs in healthy people. So with the Netherlands Donor Feces Bank, we wanted to know if this bacterium can also be found in the donors and in the patients. So we used DNA sequencing to look for this bacterium. Excuse me. And we found that this E. coli is present in a few donors, but at very low levels usually. And it is prevalent in the patients and can reach very high levels in the patients. So next we wanted to know what happens after this transplantation. So as you can see on the picture on the slide, in the middle is the PKS positive patient with this infection. So he has a lot of the PKS E. coli. And when we go to the left, you see a transplantation by a donor who also has the PKS E. coli. And we see that in that case, the colonization usually persists. Whereas if the donor does not have the PKS E. coli on the right, you can see that often the patient loses the PKS E. coli. Therefore, we conclude that with fecal microbiota transplantation, we can reduce the levels of this potentially cancer-inducing bacterium and maybe even reduce the risk of developing cancer. Thank you.
Thanks a lot for this presentation, Sam. Do you have any clue what is produced in this, in your fecal microbiota that changes this behavior of this E. coli?
You mean like in a healthy microbiota? What happens with the E. coli?
I'm just interested in whether you have a hypothesis on the mechanism.
I think the main hypothesis is at this point is that it's a sort of ecological warfare maybe, or that like other bacteria take up the niche that E. coli otherwise has and sort of drives out the E. coli.
And would in the end, would this research also result in what people might think a next step in these transplants. So now you transplant the poop, like you're saying, and people always get a little bit embarrassed when they hear that. So that at least if you understand more what's in the poop, what's doing the job, that you could do that in at least maybe also a more targeted and efficient way.
Yes, so there's research going on about this subject, and this company that we work with also is looking into which bacteria make it so that the patient cures. So they try and put these bacteria in a pill. So it's less embarrassing treatment method. But also what has been done already is freeze drying feces and put that into a capsule, which is also an easier treatment method.
Yeah, you could even sell it in the shop.
Yeah.
Thanks a lot for this presentation, Sam. It's really much appreciated. But also for you, there's a time to leave now because we need to continue with the show and make place for the next presenter. Thanks a lot.
Thank you.
And our next presenter is affiliated with the Amsterdam University Medical Center. His name is Thomas Roodzand, and he's dealing with a topic that's also close to my heart. It's called How Zoonotic Streptococcus suis Crosses Human Small Intestine Epithelium. Please, Thomas.
Yeah, thanks for the introduction. And I'd like to present my research on the bacterium Streptococcus suis. Streptococcus suis is an opportunistic pathogen in pigs that can cause zoonotic infections. And especially in Southeast Asia, the consumption of raw or undercooked pig products has been identified as a major risk factor for zoonotic infections. Already illustrating the potential Strep suis has as a foodborne pathogen. Now, while in pigs we can find many different serotypes and different genotypes, or clonal complexes of Strep suis, the majority of human infections is caused by strains belonging to serotype 2, and then especially from the genotype clonal complex 1. While in the Netherlands we have a unique clade causing zoonotic infections belonging to clonal complex 20. Due to the foodborne nature of human infections, we wanted to know if there's already a difference between zoonotic and non-zoonotic Strep suis in their capabilities to cross the human small intestine. To study this, we made a human small intestine organoid model in which we can test these infections. In this model, all the major cell types normally lining your intestinal epithelium are also present. We selected 15 strains from the 3 genetic backgrounds depicted on the slide, and we tested how well these strains were able to cross the epithelial barrier. And we found that the strains for clonal complex 1 were indeed significantly better in crossing this epithelial barrier than the strains from clonal complex 16, so the non-zoonotic strains. Using a permeability assay and also immunofluorescence stainings of the monolayers after infection, we were able to show that although Strep suis was able to translocate, the barrier function of the monolayer was still intact, which was different from previous results obtained in cell lines. So this made us for us very unlikely that Strep suis translocates via the paracellular route, so there's space in between the cells, and suggests that another mechanism is at play. Additional stainings showed that the cells invaded in the monolayer turned out to be the stem cells. Now, literature and some preliminary results we recently obtained showed that the stem cells are positive for a marker called CD77 on the cell membrane, and 2 very well-studied virulence factors in Strep suis called zoolysine and streptococcal adhesion protein are known to interact with this cell membrane marker. And we therefore hypothesize that the translocation mechanism that is used by Streptococcus suis, and especially the zoonotic strains, involves these 2 virulence factors, the CD77 marker, and all comes together in the stem cell. So to summarize, we show that zoonotic strains are indeed better already to cross the small intestinal epithelial barrier than non-zoonotic strains. And in the future, we hope to unravel the exact molecular mechanism at play by which the zoonotic strains translocate through your intestinal epithelium.
Thanks a lot for this presentation. So you also show a pig here on the screen, which of course I, being a veterinarian, like a lot. And of course it's in pigs, it's also really a big problem. Yeah. So you're dealing here with zoonotic, the zoonotic version and intestinal infection in humans, would that mechanism be more or less the same in pigs? Because in pigs there also have been debate on it, and so on the one hand we have the infection through injuries, and in the other one the hypothesis through the infection through the gut, which actually
and airways of thought. Correct. Yeah. So what I found quite interestingly is that the CD77 marker, that's a marker that's differentially expressed between pigs and humans. While in humans, for example, it's not present in the lungs, but in pigs it appears to be. So I think the mechanism could be partly the same, and I think this marker could, in both cases, play a role. But the fact that the virulent pig strains that I used were not able to cross the human barrier, while the zoonotic isolates could, for me suggests that the mechanism is not exactly the same, although parts could, of course, be applicable for both.
I understand this part on zoonotic strains being able to passage this barrier. Would that also mean that these outbreaks like we've seen in China or in Vietnam, that they are less likely to happen here because we normally do not drink the blood of pigs?
Yeah. So apparently you're known with the delicatesses of Vietnam. I think the foodborne nature of these infections is perhaps a little bit less applicable for the Netherlands. And that perhaps also explains why the zoonotic Dutch strains were actually not that good in translocating through this barrier, suggesting that in the Netherlands there's another entry point for the bacterium into the host.
Yeah. Okay. Well, very interesting. Thanks a lot for your presentation, Thomas, and answering the question is much appreciated. But we still have one candidate to go, so you need to leave the room now. And that also already brings me to the 10th PhD student. It's very nice to see that we actually end up back to the subject where we started this NCOH webinar, because we were back at Green Cities, which is, I think, making the circle nicely around. And our presenter is Marieke de Kok, who is affiliated with the RIVM, so the Netherlands Public Health Institute. And the title of her presentation, The Effect of Urban Green on Wild Rat Populations and Pathogen Transmission Risk. Please, the floor is yours.
Thank you. Yes, so my name is Marieke de Kok. I'm a PhD student at both RIVM and Wageningen University. And my research is about zoonotic pathogens in wild rats in general. As you have already heard today in the other presentations, urban greening has a lot of beneficial effects. So we should definitely make cities more greener. However, the potential negative effects are often overlooked. For example, by making cities more green, you also increase biodiversity, which is a good thing, but thereby you also get more wildlife into the cities and potentially more contact between humans and wildlife. And this can increase the risk of zoonotic disease transmission to humans. In my talk, I specifically would like to focus on wild rats. Wild rats are abundantly present in urban areas, and they can carry various zoonotic diseases. I'm sorry. And a previous study in Amsterdam showed that wild rats, or at least the complaints of wild rats, have been correlated with the amount of urban green that's present in a certain location. So that means that the more rat complaints there were, the more green those locations were. But rat complaints are not, there are complaints that people file when they have rat nuisance in their neighborhood, but they're not one-to-one translatable to actual rat abundance. So in my study, we want to look if you can see this correlation between urban green and rat abundance. So we have 3 different location types. We have urban parks, which are highly green. Then we have green residential areas with more than 40% green, and gray residential areas with less than 20% green. Per location type, we have 16 different locations, all randomly selected. And there we capture rats in a systematic way so that we can compare the relative rat abundance between the different locations. And the rats that we capture, we also collect them to test for specific or various amount or various number of pathogens, like bacteria such as Leptospira and Bartonella, but also antibiotic-resistant bacteria, tick-borne bacteria like Rickettsia and Anaplasma, viruses like Seal orthohantavirus and hepatitis E virus, and parasites like Toxoplasma gondii. And by doing this study, we hope to see whether there is indeed a relation between the amount of urban green and actual rat abundance, and between urban green and pathogen prevalence in these rats, to ultimately say something about the risk for public health. Thank you.
Thanks a lot for your excellent speech. A question is, because we know that in several habitats, rats and also mice are becoming more of a problem. Also because at least dealing with this problem is getting more troublesome. Do you in your project also envisage to try to come up with solutions? So once you've found an association, so it becomes, the populations grow, how the human population can deal with that?
We try to. It depends a bit on what the results will be of this study. But if, for example, we find more rats in urban green space, then we, together with municipalities, would like to see how can we maximize like the urban green positive effects and minimize the negative effects of rats. So maybe you have specific type of shrubs or trees that are more favorable by rats, and then you can maybe switch to other types of green.
Thanks a lot. Thanks a lot for your presentation and your pitch. Thanks a lot for answering the question. And you also need to leave the room now, because I need to ask now the co-moderator of this session, Maarten Hoek. But so you need to switch rooms now. Who is going to do a very important task, because now you've heard all 10 PhD students giving their pitch, and Maarten is now going to tell you how you can give your vote to the best one or the favorite one. Maarten, please, the floor is yours.
Arjan, wow, what a talent we have, right? Within NCOH, so proud to be part of it. But before we move on, let us give you a token of our appreciation. It's not a pig. You said you love pigs in the presentations. We can't bring that in. So we give you a bottle of bubbly pink stuff.
Thanks a lot, Maarten. It was not needed, but thanks a lot.
Thank you for your role so far. We'll get you back, of course.
Sure. Please.
So now we move to the voting. Now you can vote to the PhD student who gave the best pitch or gave the most interesting research. What is on the line, what they can win, is this very valuable cup. It's almost as valuable as the Henry Delaney Cup from the European football. I have extra security, and it's nearly invisible online. Sorry about that. It's glass, but it's very pretty. And we also will give out the stress balls to all the stressed out PhD students. So if you want to vote, please now go on your computer or on your phone. Oh, before we go, sorry, on the right bottom, I think it's here, press the subscribe button for our NCOH channel. Right now we go to the www.menti.com, where you can vote for the pitch of your preference. So press in the code 35779934, and then you can vote for the PhD student which you think presented the best or had the most interesting research. You can vote for poop transplant, or you can vote for bird tracking. You can vote for macrophages, as in pigs from Thailand. You get 1 minute in total. So go to menti.com and press in 35779934. And I'm just checking. Mike, are we getting the first votes in? Are we getting the first votes in, Mike? No, very good. First votes are coming in. So please keep voting. On Ayla from de Weur, on Remy from UMC, Kitty from Utrecht, or Jurian from NIO, Joanne from Leiden, Michelle from Erasmus MC, Esther from Radboud, Sam LUMC, Thomas Amsterdam, or Marike from RIVM. Who is going to be? Who is going to win? The votes are coming in. I see the excitement by the panel. It's really growing, really growing. Yes. Do we have a clear winner yet or not yet? No, it's undecided yet. So your vote counts. Please, please, please vote. And if you've already voted, press the subscribe button on the NCOH channel so you stay up to date with our webinars and other news events. It's going really well here. It's a tense. Oh, it's a tie. I hear it's a tie. So please keep voting. I didn't know it was going to be so exciting. It is really exciting. Oh, we're slowly going to have, are we going to have a winner? Somebody taking the lead slowly? Ooh, I see. I think it's closed. Is the voting closed? We're going to show the winner right now. And the winner is Sam Noy from Leiden University Medical Center. Sam, please come inside and take your cup, your winner's cup. Sam, please. Sam! Yeah, he's still busy on his PhD, I guess. He's really dedicated. Always researching. No wonder he won. He's coming. We took him away from his research. Sam, on behalf of the NCOH, the much prestigious annual NCOH Best Pitch Award goes to you. Congratulations.
Thank you very much.
People have voted. Do you have an award for the voters? Oh, you have to come from the screen.
Right.
Oh, camera has to move. Yeah, very good.
So, first of all, I'd like to thank everybody who voted for me, of course. I'd like to say thank you to my amazing colleagues in Leiden who made this work possible. Thank you to my parents, my family who may also be watching. Also, thank you to my employer for already providing me with the vaccine against COVID, which is doing its job right now. And yeah, I'm glad you liked it. And hopefully the paper will come out soon, so we'll be able to read all the details when you have the time.
Very good. Congratulations. Well done. We hope to see you back next year. Here's also a stress ball.
Thank you.
For the stressful moments. Good luck finishing the study. Or the PhD, sorry. And then we give the word back to Arjen.
Thank you, Maarten, for doing this great job. And congratulations to you, Sam. But also, I would like to congratulate the other 9 PhD students, because I think they also have done a marvelous job. So, because for me, it would not have been easy to vote for a winner. So thanks a lot. And I would like to thank also the people organizing this Netherlands Center of One Health. I think we've seen an excellent program with initially, at least for me, an exciting topic on greening of cities. I think we will hear lots more of it, because we all like to live in a green environment, but many of us are not aware of the risks associated with it, and I think many of them we would need to take in a responsible way, and research in that area is very important. I would like to thank the core team of the NCOH webinars who is organizing this. Maarten, you've seen, but also Maaike, Erika, Jovanka, Paul, and Mary and Marie, who are doing a great job. I'd like to thank Matthijs, our technician, and his co-worker here for doing that job. But I think most of all, I would like to thank you, participants. Oh, I forgot. I also need to thank Dominique, who I think did an excellent job in the Energizer, but also the nice singing before. And I'm sorry that I don't know the name of the person playing the ukulele, but I really have a soft spot for people who play the ukulele. So I would also like to thank him a lot. And also, and that's, I think, most important, we make these webinars for an audience. So you, as participants of this NCOI webinar, I would like to thank you a lot for participating, for your contribution in providing questions, for your contribution in voting. So because that's stimulating, I think, for the PhD students. We have seen 10 excellent presentations who did it in one slide, one person smuggling a bit, but we don't pay attention to that. So you see that there's a lot of talent present in the NCOH, and that also makes that the NCOH will have, I think, a bright future. And with that, I would like to thank you, and I hope to see you all at the next NCOH webinar, which will be on emerging zoonoses and how the contagious jump between animals and humans occurs. So thanks a lot for your attention, and hope to see you soon. Bye-bye.